Okay, so good afternoon, everybody. A bit of a different host today. You've got me, Paul Shaw from James Lange Institute. First of all, can I welcome you to today's APGC seminar. Today we welcome Professor Yiliang Ding from the John Innes Centre down at Norwich, a place that so many of us will have had collaborations and interactions with over the years. And Yiliang has been a group leader at GIC since 2014, focusing on RNA structure, functionality and cells. She also holds honorary positions at the Babraham Institute and University of East Anglia. Yiliang completed a degree in Shanghai in 2005 and moved to GIC to undertake her PhD, which she finished in 2009. She then went on to postdoc at Penn State in the US until 2013, and then a move back to sunny Norwich. Yiliang has received several prestigious awards and grants over the years, such as the received David Phillips Fellowship, 3 ERC grants, and the Royal Society Faraday Fellowship. Yiliang is also one of the 9 recipients of the 2024 Blavatnik Award for Young Scientists in Life Sciences in the UK. So without further ado, I'm going to hand over to Yiliang. Yiliang is going to talk today about decoding RNA language in plants. So Yiliang, over to you.
Yeah, thank you so much, Paul, for the introduction, and it's very honored to talk here online. But hopefully, after my talk, there are a lot of interesting people willing to collaborate with us. I think there's a 2 institute collaboration is quite extensive, so it will be really nice after my talk. Someone of you actually willing to working on RNA a little bit with us. That would be great. So today I'm gonna basically talk about several tools developed in my lab in the past pretty much 11 years, and I give you idea how many kind of tools currently on the table for you to use in your system. So as actually you see in the central dogma, we kind of focus, a lot of people focus on transcription regulation, chromatin architecture, and nowadays AlphaFold had a very kind of a large amount of a protein structure there. So in the textbook, you always see messenger RNA as a very simple single line. It seems like a messenger RNA don't have any structure. So then what we actually, through the past, I think decades or even more longer than that, people find that if you dope any kind of RNA with different sequence lengths, with different actually sequence content, you will always see a very beautiful stem loop structure like a GC, GU, AU, GU base pair. And sometimes for nucleic acid chemistry, they will actually also find that it's a very complicated structure like a pseudonote, triplex, also G-quadruplex tertiary structure. So the question for years is really about how you can actually really gather these structure information inside the cell and how these structure inside the cell really have a certain role towards different RNA function. So this is really started from the beginning of when I started my own lab. It was the time about 11 years ago, a group of people from globally actually made an effort to develop this kind of a method called in vivo RNA structure profiling. So the idea very simple. We use a kind of a chemical compound called a dimersa sulfate. It's very small, so it's easily penetrated into the cell, then modify the RNA base. So when A or C nucleotide is a single-stranded, this method group will actually tackle to the Watson Crick phase, where if you have a CG pair, this method group will not get on. If it's don't form CG pair on the Watson Crick phase, this method group will actually attach to the phase. So once it attached with this method group, of course, during reverse transcription, you will actually see a reverse transcription stalling at a specific nucleotide. This nucleotide got modified. So if a lot of actually reverse transcription stalling at this specific nucleotide, that means this nucleotide are likely to be modified by this dimersa sulfate. So likely this nucleotide is tended to be more single-stranded. So then we actually combine Illumina sequencing using single-stranded DNA ligation, put adapter here. So then we'll be able to capture transcriptome-wide every single nucleotide on every single gene, their single-stranded probability. So we can measure something called stalling number then calculate a chemical reactivity. So if you have a high chemical reactivity, that means this nucleotide tended to be modified more heavily. So that means this nucleotide tended to be more single-stranded. So if it has less stolling, that means it's likely to be double-stranded. So you have a low reactivity, so representative of that. Then through years, nowadays, we can using this dimer sulfate to probing both AUCG, not only A and C, and also through changing the reverse transcription reaction rather than 1 hour, we can add a magnesium chloride. So what actually can let a 3 hours reverse transcription, let this reverse transcriptase to bypass this modification site, then introduce a mutation. Again, using mutation profile, we also can measure how much chance, how many nucleotide was modified, then generate a chemical reactivity. So keep in mind that either is a stalling or mutation profile can capture this kind of a single strandness of RNA. So the other chemical we're using in the lab, rather than the target to the base, it's actually isolate the single strand RNA on the 2' hydroxyl group. So again, it will make a block on the RNA. It also can be detected by either reverse transcription stalling or mutation assay. So this is how this method is working. So far, we actually go back to the textbook. It's really about 11 years ago. I was really ambitious, which we kind of update a little bit on site. So again, you see from the RNA burst, when you're doing the transcription, splicing, polylation, then mature mRNA going through translation, RNA degradation, even the microRNA mediated RNA cleavage, you can see in the textbook, every RNA was drawn as a single line. So the question really is, whether or not they really as a single line or they do have a certain structure which associated to their function. So I want to go that very deeper. I actually point out the model system we're still working on Arabidopsis. But however, through years, in the past 11 years, we're working on different kind of diverse of a plant, which we try to understanding, how the structure functionality to individual species. So it's not only limited in the wheat now, we also can doing on the barley and also brassica, which if anyone in your institute interested to work on with us. So basically, this is a slightly update on how much we understanding, but I will actually go more detail on specific case. So what we find is that the nascent RNA, so pre-mRNA, do have a certain level of structure, which a certain structure, especially two nucleotide upstream 5' splice site control the splicing efficiency. Also, we can find these polylation specific structure feature, which corresponding to the RNA maturation. We also actually can find the RNA had a different motif which regulate the translation efficiency. Also, structure motif actually contributed to the RNA degradation. Of course, we also can find a structure do exist in the microRNA substrate and messenger RNA, which actually regulate their interaction with microRNA. Interestingly, through years, we accidentally find if you have these kind of a tertiary structure, which I'm going to talk about in detail, called the G-quadruplex tertiary structure, itself can form RNA-driven phase separation. So people interested in protein phase separation, yes, RNA do itself can also form phase separation. So today, I really want to focus is this kind of a really cool RNA tertiary structure, which called a G-quadruplex. So what is RNA G-quadruplex? So if you're looking for the guanine nucleotide, you will see Watson-Crick phase, Hoogenstein phase. So if you have a 4 guanine nucleotide together, it will form these kind of GGG plane structure. So you will see hydrogen bonds form together, so you actually have a relatively stable 4G kind of plane structure. So if we have 2 layers or 3 layers in human, maybe 4 or 5 layers. Also in majority crops, you actually have 4 or 5 layers. So then in the sequence, you will see these kind of 4 repeat of G2 or G3. So go through the sequence, you have a GG loop sequence, GG loop sequence, then GG loop sequence, then GG. So this sequence, if you search in your transcriptome, it's everywhere. So what kind of things actually trigger this kind of G-Cordoplex formation is when you have an ion like potassium, sodium, nitrate, even calcium can stabilize this kind of tertiary structure, make it super stable. So if lithium, under lithium condition, because lithium is very small, so it won't hold in this structure. So people in the nucleic acid chemistry, research field, they found that this kind of structure is actually likely to fold very stable in vitro. So the concept again is if it's a fold very strong in vitro, so can we know whether or not it's truly folded in vivo, right? So this is really based on a very interesting observation in our chemical probing. So if you still remember this shape chemical, which is acylated 2' hydroxyl group, when we actually find is when this G-Cordoplex tertiary structure folded in the potassium condition, every last G of every G repeat is exposed their 2' hydroxyl group more preferentially than the rest of nucleotide. So because this preferentially modified by this shape chemical, so when we perform a reverse transcription stalling assay, you will see it's preferentially stalling at the position which is this last G position. So then through deep sequencing, you will see a sequencing pattern which is uneven distribution of reads count. Interestingly, is if you put this G-Cordoplex, the same sequence in the lithium condition, you lost this kind of a preferentially modification of this last G of every G repeat. So when you do the reverse transcription, you will have more even distribution. So when we observe this kind of phenomena, we thought, okay, shall we actually use that as a standard to, measure, set up a standard so later on we can compare with the in vivo profile. So what we did is we using a chemical, by physically chemical validated kind of a G-Cordoplex candidate. So this is a G-rich sequence. So then put in lithium, you will see it's more even distribution. But if you put this sequence in the potassium, you can see it's very skewed on the G, last G of every G track. So then we thought, okay, then we did a in vitro transcriptome-wide folded standard and a in vitro transcriptome-wide unfolded status. Then using our in vivo probing to compare with folded and unfolded, then we will know whether or not the in vivo is folded or not, right? So then what we did is we then probed the in vivo. Then you can see this in vivo G-Cordoplex is very similar to the potassium folded status. It's very skewed distribution rather than even distribution. So we know this G-Cordoplex is folded in vivo. So because we do transcriptome-wide, so we can measure all the transcriptome-wide G-rich sequence, whether or not they truly fold into this G-Cordoplex, motif. So you can see a distribution close to 1. That means most of the G-Cordoplex sequence is folded into this structure. Interesting, like rice, also wheat we're doing now, is actually you can see a much more stronger folding status comparably, of course, because it's also GC-rich kind of a transcriptome. So you can actually see rice have a more stronger, and most of the rice fold a much more stronger status of G-Cordoplex. So the question really is, why this structure really have a functional, what do they really do in the cell, right? So we actually, once we got all these transcriptome-wide, we immediately do the geoanalysis. Not surprisingly, you have all these stress response through also water deprivation, also RNA processing, geoanalog. So my postdoc said, okay, how about we just put it into one of the stress? The simplest one is actually just to put it into temperature. So we select a response to cold. So again, we actually put the Arabidopsis plant into cold 4 degrees for 2 hours, do all the probing, chemical probing again to compare with in vitro folded and in vitro unfolded standard, just to try to see what happened of this motif in the cold, right? So what we actually found is some G-Cordoplex motif in the transcriptome previously is not folded. It's very similar, 22 degree, similar to the lithium unfolded status. But when you put the plant into 4, you see it's very similar to the potassium. So that means this G-Cordoplex is induced in response to cold. So again, if you measure the whole transcriptome, why the G-coded plaques, what happened in the code is you will see a shift to a much more strong folded status. So that means globally, all these G-coded plaques folding strength is increased in response to 2 hours of code. So then my postdoc said, okay, what do they do, right, if you have a G-coded plaques form? So at the very beginning, we think a G-coded plaques may be responsible to suppress translation, but we didn't see anything after 2 hours cold treatment. So then my postdoc did this kind of RNA decay measurement, which you transcription arrest by a chemical. Then through the time course, you measure the RNA decay rate. So every RNA-seq, then you generate these kind of decay curve. So we did it in the 22-degree and 4-degree. It's a very complicated figure. I'm just go very simplified. So what we can find is those G-coded plaques induced in the 3 UTR of RNA, when in the 22-degree, it's actually have a very fast degradation. But once you move to the cold, these cold-induced G-coded plaques genes is actually have the most stable kind of phenomena. So that means these kind of cold-induced G-coded plaques in the genes in their 3 UTR is tended to stabilize the RNA much more stable in the 4-degree condition. So of course, we want to validate whether or not that's the case. So we're using fused report assay. So where we put a luciferase, then put a 3 UTR contain this code-induced G-coded plaques sequence. And we actually perturbed this G-coded plaques motif, just basically mutate a couple of Gs to A, disrupt the capability to form G-coded plaques. So then we put this 3 UTR mutated G-coded plaques into this report assay. What you can find is because this G-coded plaques only induced in the cold. So in the 22-degree, you don't find that the stability decay rate has any change. So the RNA degradation quite fast. But in the cold, in the wild type, the RNA get very stable. But with the mutated G-coded plaques, you see a decrease of a fast degradation event. So that means this G-code-induced G-coded plaques do have a control of stability in the cold condition. So really, we are, plant scientists. We really want to see whether or not there's any phenotype. So my postdoc immediately actually ordered, a tDNA insertion line, which is a knockout line of this gene. So you can see in the 22-degree, there's no difference of, plant root difference. But this genes in the wild type, normally Arabidopsis columbia, you will see after 4 weeks cold treatment, you will have a shorter primary root length. But in this knockout mutant, it's showing insensitive, so you have a longer root. When we complement the whole gene with 3 UTR with this code-induced G-coded plaques, like normal complementarity, you will see it's back to the suppression of root length in response to code. But when you complement the whole gene with 3 UTR, only have this G-coded plaques, some Gs mutated to the A, you will see it's getting longer. It feels like similar to the knockout mutant because in this status, this RNA going through the faster degradation. So that's why it's similar to the knockout. So you do really see if you perturb, even non-coding region, a structure motif, you can have a huge impact towards actually their code response in the plant. So when we actually doing this G-coded plaques work, we actually just a very nice publication in Nature published these 1,000 plant transcriptome. So my postdoc said, if G-coded plaques really sensing code, is there any actually selection of G-coded plaques motif through transcriptome of these 1,000 plant species? So what we did is we actually predicted all the G-coded plaques density. So this is a 5 UTR, 3 UTR. Density crossing every single one species, So then we collected all 19 climate parameter. Surprisingly, very surprisingly, the G-coded plaques density in both 5 UTR, 3 UTR had a very strong anti-correlation with the temperature parameter. So that means if these plant species tend to live in a cooler area, so relatively cooler area, their whole transcriptome tend to have more this G-coded plaques motif crossing their transcriptome. So we are currently trying to investigate how, how these selection really do, what happened to these species actually located in the cooler area, whether or not they have a really lot of in vivo folded, G culture plaques. The interesting part is when we in the RNA Society meeting, we found our topic with another group in Harvard, they actually doing similar things. So in the human, G culture plaques is not formed under 37 degree, okay, because it's thermodynamic too high. But however, when they push a little bit of stress, like UV light, also cold, they can find these G culture plaques also like a kind of a form stably inside a cell. Interestingly, they also find the majority is located in the 3-UTR genes, which try to make this RNA much more stable. So this is really kind of aligned with our plant kind of observation. So this is giving you idea is likelihood this kind of, G culture plaques motif is a universal stress sensor. So now we actually also build up a data resources of all the G4 atlas. So you basically can see your gene, whether or not this gene is actually have any G culture plaques formation in your data set. So everyone is welcome to visit our database. So currently we host quite a bit of rice, Arabidopsis, even human data. We also gonna host, wheat data into that. Maybe someone interested in working on barley of G culture plaques, we can also, get more data set on that. The other kind of interesting motif, which we call I motif, which is opposite to the G, rather than the G rich repeat, you can actually see this kind of a CCC 4-time repeat actually form these kind of also beautiful tertiary structure called I motif. So this is also we made some effort towards how to predict this kind of motif. Currently, we're still actually working on method to actually probe it in vivo, but this is also a database which everyone can actually interesting to see whether or not you have any kind of link to this tertiary structure motif. I needed to point out this motif is actually pH sensor. So low pH can really strongly induce this formation of a motif. So another technology which I want to emphasize today is really a transformative understanding of RNA structure. So this is one sequence, how many possible in silico conformation you can have. So it's very diverse. It's not like protein structure. RNA, every single RNA sequence can fold a very diverse conformation, right? So then the question for years really is how to obtain, individual RNA structure conformation inside the living cell, rather than this, in silico prediction, whether or not we can gather the real kind of conformation inside the living cell. So today I'm going to demonstration this transformative technologies through a very famous long non-coding RNA called QL. So if most of people working on plant, you will know plants require a vernalization stage to be able to flowering. So during the plant vegetative stage, when they sense the cold, they will suppress a very important gene called FLC genes. So once you suppress this gene, the flowering will start. How to suppress the FLC gene is through its antisense long non-coding RNA called QL. So this QL will be cold induced, and this QL long non-coding RNA will associate with the chromatin of the sense gene, so suppress this FLC expression. So it's really the beginning when I started lab, we have this famous lady, Caroline Dean, right? So she said, can you use your chemical probing to really probe this QL structure? When I look into the QL, it's very difficult because you have so many different, isoform. So if using traditional Illumina sequencing, you won't be able to know the structure is coming from which isoform. So we really specifically design a method which can able to, for us to actually probe all the structure of individual isoform. So this is the methodology. Again, it's a plant. You can chemical treat it. All the single-stranded nucleotide of all the RNA will be modified. So you can actually basically using PCR to pick up the gene or isoform you wanted, then generate the mutation profile of all the cDNA. Again, with double-strand DNA, all the double-stranded of these mutation will representative of single-stranded of every single molecule. Again, you can put into, pack-by sequencing for single molecule sequencing. Also nowadays we're using nanopore quite a lot. So each hi-fi reads is a mutation profile representative of structure information. So each reads can actually come up with a structure conformation. So if you're sequencing, 1 million reads, then you have 1 million structure. Then we can cluster the structure, then we will see how the structure conformation it is. So this is one of the major QL isoform. You can see after our single molecule structure profile, you can see there are actually 3 major clusters. So this is very high resolution. So once we got the data, so then Caroline said, can you put in the code, see what happened? Remember, previously, all the RNA structure is ensemble structure, is average conformation, right? So how about, single molecule resolution? So we put into the code 2 things. We actually found some conformation going down as a proportion-wise, some conformation proportion-wise going up. You do see a very code-specific structure conformation. Right. So with this resolution, we really can monitor what happened to every single molecule inside of the cell in response to a stress. So then we're choosing a very highly dynamic area, which we just basically make a, 4 nucleotide mutation, we make this structure super single-stranded. So, and we also validate through single molecule profile. So once you make a QL very single-stranded, so it's associated with chromatin stronger. So once it's associated with FLC chromatin stronger, you suppress FLC expression. Of course, you can get early flowering. Okay, so this is giving you a sense once you have a very clear resolution of a structure, you can identify the key site which is important to the gene function. So this method now is everyone can work on the bench within 1 month, you can actually get your structure beautifully. So it's also another part which is more dry bench, which I want to mention, is actually newly developed in my group. Apart from all these experimental kind of, platform, really about pretty much 5 years ago, when I handling so many structured data, right, in the cell, we really have a question is how we possibly to understanding, across RNA, let's say 1 kb or 2 kb, which part of a structure motif is really corresponding to certain function we're interested in, like a translation efficiency, transcription regulation, or RNA maturation or splicing, right? So then this is the moment I actually think, okay, we should actually adopt a little bit, AI. So I think this part is really familiar to everyone. Is nowadays everyone kind of using ChatGPT or other platform like DeepSeek to draft your email, data analysis, also reading paper, right? It's basically like everyone now using that. So what actually really created ChatGPT is something we call foundation model. It's also called a pre-training model. Also we call large language model. So if you think about it, if traditionally, people talk about machine learning, deep learning, right? It's basically is ask a baby, if I give you 1,000 Shakespeare poems, can you write a poems in Shakespeare style? A baby will cry because, 1,000, Shakespeare poems is hard for a baby, right? So what a really pre-training model or foundation model, or we call, large language model do is actually training this baby with all the possible knowledge like literature, classics, history, everything, right? So in that way, is you build this baby already become a PhD in literature. So well pre-training model, right? So once this PhD receive these like 1,000 Shakespeare poems, of course, the PhD likelihood is actually he will actually write a style, Shakespeare style poems, right? So this is a similar when we think about how we basically generate a pre-training for the RNA language. So give you a little bit more detail towards how different AI platform, right? So think about machine learning traditional 15 years ago, people really rely on that is basically you have a data set, Okay, you have like a cat figure photos, dog photos. Then you tell which part of feature is belong to cat, which part of feature is belong to dog. So then you create some model which can predict whether or not it's cat or dog, right? So basically, like if you have a cat photos, you can give a feature like a fur color, fur length, whisker color to actually describe this cat. So the model have some information, then they will actually learning stuff. So deep learning is differently. So you have to create a lot of photos of cat, a lot of photos of a dog. So be able to learn the feature itself. But that means you have a large amount of a data set you require to do that. Okay. So what a foundation model really do? You create an animal database, all the photos of that. Don't tell the model, what it is. Let it itself to learn what is a cat or dog or whatever. So you give a little bit of fine tuning, like 10 photos of the cat, dog. You will generate something called a cat dog classification model. Give a little bit of data set, like 10 photos of a chicken duck. You can get a chicken duck classification, and the tiger lines, you can get that. So that means I can have a pre-training which I don't need a label, which means I don't need to tell you anything. I just give you a animal kingdom photos, let you to learn all these kind of feature. Then later on, I just need to give you a little bit of supervision. Then you will be able to create, what I wanted. So the similar way is how we did that is we generated something called a plant RNA foundation model. What we did is we sending these 1,000 plant transcriptome information with huge diversity their sequence, then put into this large language foundation model. So what do we put into it for training? We put into it as RNA sequence, RNA annotation, where is the 5-UTR, where is the CDS, where is the 3-UTR, and we also put in silico predicted structure motif as information. So we just let it self-learn through 2 months in 7 GPU, H100 GPU. So we actually got what it learned. So what it do is actually try to understanding evolution kind of trends, which the sequence coordination, which our eyes couldn't capture. So once we have this pre-training model, again, as I said, then you do some fine tuning, right? So what we are interested in is, okay, you have a 5-UTR, you have so many structure, you have so many sequence. So exactly which part is important for, translation, right? So previously deep learning couldn't do, because if you think about it, you have a 300 nucleotide of 5-UTR. It's very hard because you have 300 variable, but your translation efficiency is only one value. How you actually learn, 300 variable to do that. So majority deep learning kind of a methodology is harder to do any regression learning on these kind of data set. However, now what happened is we give a 5-UTR sequence, it converts into pre-training. So every single 5-UTR sequence crossing 10,000 genes, right, or 20,000 genes. So each sequence will have a token number. You have 52 million metrics to measure individual 5-UTR sequence. So rather than just 300 nucleotide, you have each 300 nucleotide behind it has a 52 million metrics to measure that. So then when we actually put a high translation 5-UTR, low translation 5-UTR, you have a correct label of a translation efficiency. Now putting into the foundation model, and we're actually starting learning stuff because of these pre-training already done. So what we have set is an input with all the 5-UTR sequence. They're labeled high translation, low translation. We also can chiffering the label and do a parallel learning, which is called a random learning. So this will give you learning as a 50% of learning chance, right? Random learning. So then each foundation model after deep learning, you will come up with something called attention score. So we subtracted the learning from the true data and subtracted the random learning data. We call something called attention matrix, contrast matrix. So attention is something to measure every single nucleotide, how important to its translation efficiency. Okay, so then we end up, a Rabbit-Arcis, rise with this data set. So we said, okay, we average all these 10, 20,000 genes just to see what is the general trends of these attention. So you can obviously see, no matter it's a rhabdosis or rice, you will see globally attention contracts getting higher close to the ATG, right? So you will see, no matter it's a sequence motif or structure motif, when it's close to the ATG, it's important for translation efficiency, okay? So when we're looking closer to a certain area, like close to ATG, you will find something called a Kozak sequence. So keep in mind, I never tell the machine there's something called a Kozak sequence. It learns from itself, right? How amazing it is, it actually can learn, something we actually experimentally identify as a Kozak sequence. So anything else it can learn. So then we overlay our structure motif correlated to these attention contracts matrix. So we just try to find which part is important, which structure. As I said, if I have a 300 nucleotide, which part of the structure is really corresponding to translation. Now I have attention contrast. I can overlap with my structure motif, so I can identify exactly which part on this RNA, which motif is important for its own translation. So then, of course, I can run bioinformatics cluster, just which is a high translation motif, which is low translation motif. So not surprisingly, we can find these low translation motif with a high GC content. You will see very stable 4-stem loop kind of a GC motif, which enriched in the low translation motif proportion. You need to think about it. Previously, everyone talked about high GC correlated to low translation efficiency. But exactly what that high GC looks like. Now with our foundation model, we can really see this kind of a specific motif at every individual's gene. So again, we interestingly actually found that these are high translation motif, right? So RNA is not a remain complete single line. It do form certain structure, but relatively to the low translation, you have these AGCU kind of repeat sequence, relatively weaker stem, but you do have a structure motif. So interestingly is when we actually saw that, we searched the literature in Drosophila, there's truly a case is they found in the 5-UTR AGCU repeat sequence is associated with a high translation efficiency. So then we actually found that it's actually a global effect. So if you perturb low translation motif to high translation motif, you see you can actually doing report assay easily find that it's actually can getting higher. So if you actually put high translation motif changing to low translation motif, you see you can actually suppress the translation, right? So this is how identify. So every single gene now you can know exactly which kind of a motif corresponding. If you do the perturbation, you can actually change its translation efficiency. So then, if you still remember RNA quadruplex, that motif I told you, so in the 5-UTR, it also can suppress translation. But over 65,000, which one is really suppressed translation confidently, significantly, right? So we can identify 21 low translation associated RNA quadruplex. So of course, if you're changing a little bit of G to A, abortion the G quadruplex formation, you can see I can actually have a high translation. Okay, so, we are doing AI, right? So my bioinformatician said, because we're doing AI, can we actually tune in the gene expression as what we want, right? So we can actually really doing design. So again, if you still remember this overall attention contracts, score, where you see close to the ATG, no matter sequence or structure motif is more important, but outside of like a 200 base, it doesn't really actually matter any longer, right? So my postdoc said, I know G quadruplex is suppressed translation, so can I do a simulation? So if I move this G quadruplex from the ATG to the end of beginning of a 5-UTR, so I can simulate it. He said, when I move this G quadruplex to the beginning of 5-UTR, the 5' side, so it will lost its translation suppression role. So it doesn't really matter to the translation suppression any longer. So this is a computational simulation, right? You really want to know whether or not it's working. So we did a reporter assay. So we actually, this is a native G quadruplex. We identify inference translation of this gene. So if we move it close to the ATG, it suppresses more. Completely attached to the ATG, completely suppress the translation. So if you move out, it's actually increase. Move out, more increase. Move towards the beginning of a 5-UTR, it's very similar to the mutation. So you lost the suppression role. So you basically can design, a 5-UTR, a structure motif at any position of your 5-UTR to be able to quantitatively control your translation efficiency. So why we call it a foundation model is because we have a foundation model, then it really depends what you want to learn. You can learn translation efficiency, you can learn degradation data, you can learn RNA binding data, you can also learn RNA modification. So then we can also do similar things as decoding, like interpret what actually the machine is learning. And of course, you can actually do a lot of design for the RNA as what you wanted, right? So the other thing I want to mention, because still a lot of people working on animal, we also parallelly have another powerful 1000 animal transcriptome pre-training model, which also including all the structure parameter with sequence annotation, also structure information. This is a slightly different than the transformer normal one. You think about the transformer is like ChatGPT. This is called the MOE structure, which is much more powerful, similar to the DeepSeeker kind of a platform. But truly, we do have an animal RNA foundation model now available, can do all these kind of cool things with functional RNA motif identification in the cell. So I just needed to introduce another things which is actually currently on the way is we're using ensemble plant 157 plant species. They are well annotated genome. Okay, again we did a self-learning pre-training, also annotation with promoter exon intron information. Then we put all these information over 157 plant species to this kind of a transformer contact. We generate a plant DNA foundation model. Give you a little bit of slightly see the DNA, but we actually have more things on the way. So if I randomly in Arabidopsis or rice, we randomly select a promoter region 200 nucleotide, exon 200 nucleotide, intron 200 nucleotide, terminator 200 nucleotide. So normally lead sequence is really independent. You won't be able to understanding there's any actually things come out, right? So after our foundation model, you can really see sequence itself clustered together. So the model really can tell you this sequence is from promoter or terminator or intron or actually exon. I needed to point out exon part, this part is CDS. This is a 5-UTR, this is a 3-UTR, which means after the pre-training, every single sequence of this 200 base pair from everywhere, they will have a 52 million or metrics to measure that. So sequence itself is not a sequence any longer. It's a sequence with 52 million metrics associated with that. So when you actually compare all these metrics, you will be able to know this 200 base pair is coming from exon, intron, or terminator, or promoter. So this is powerful towards nowadays, we actually, based on the evolution, we can actually really through these pre-training step to understanding the sequence itself, some hidden rules behind it as actually what it come up with learning. So the other platform I want to mention, I hopefully after my talk, everyone gonna start to try that. So everyone notice is if you put a question, in the ChatGPT, you always, especially plant, related, very specific professional kind of a question, you got a wrong answer. For example, if you ask what is FT, flowering time genes into it, like FT, there are so many things comes out. So that's why at the very beginning, we thought, okay, we build up, a foundation model for RNA, a DNA. So whether or not we should build up a knowledge kind of things for the plant research. So we thought, okay, we should build up something which is continuously learning. Not only one, like if we just give a 20,000, publication, then it's only learn 20,000, then it's useless to you because 2 months later, this 20,000 will become, 30,000 literature. So how are we gonna keep it up? So we build up something called automated scientific knowledge graph construction. So what it means is if you continuously feed in the new literature, it actually will kind of learning all the knowledge. And it will update continuously when you feed in new knowledge, then it's continuously update this kind of knowledge. It's like a scientist rather than a database. So this website is actually available. Everyone can register, called Plant Science AI. So how cool it is. So this is what a ChatGPT or Llama or Cloud will give you is FT, the main target of constants, right? So it's plant people will know because FT is a gene called flowering time. CO is actually constants. But however, this platform don't know because it's very diverse knowledge. It's not a build up for plant scientists, right? So then that's the question. Now we put it into our plant science AI, right? Because we learned the literature of FT, flowering time, constants. So now we can gather the correct answer of what is FT means, what is constants means, right? So this is one benefit with plant science AI. So if I show you how you actually get, you can answer any question. It will show you how many actually the knowledge in this whole database, of course, is continuously built up strongly. So you basically can see all these information. Then based on all these links, you can actually collect it. Each one will give you how this information comes through, and you can have all the literature, which you can actually go to search it and actually understanding far more better. Every single dot here is called knowledge node. So for example, FT is a node, and the constant is node. So you basically all the knowledge connected together, you actually try to find a lot of link together. So that means the answer we give to you is based on the link we actually found. So you can tracking back how the answer really generated. So if you so it's actually where you actually putting on, I think you will actually see, I think it's just to go down next one, sorry. So if you actually see where you have knowledge graph, there's something called a show knowledge graph. So for example, flowering time, you can have so many literature already about it. So each node, you can actually select that. Every single node is connected together. So basically you will know all the information about the gene you use. It's good for graduate students, postdoc who just started in the lab, who want to understand the whole thing. So you can basically put your whole endnote, every lab put that. You can also build up a very specific knowledge for all the connection we have. The reason we have so many FLC is because Caroline Dean kind of like give us 400 literature on the FLC. So that's why we have a heavily data set. But the thing is actually continuously update. So every single knowledge can actually go ahead. So at last, I want to acknowledge my group is getting bigger and bigger, and also our collaborator. I need to say is my lab is very lucky to have a lot of collaborators like Caroline, Ke, and Zui. All these people actually help us to try to build up a lot of nice tools, so be able to contribute to the community. And I hope after my talk, more people want to, using our platform and either probing RNA structure or doing all these fancy, RNA AI work. So thank you so much.
So thank you, Yiliang, for a fantastic talk there. Just open the floor if anybody has any questions they might want to ask. If you either raise your hand and Raman will allow you to speak, or post it into the chat box there. I guess a quick question, Yiliang, maybe just to get started, is where do you see your work? Where do you see AI progressing now towards the stuff you're doing, and where do you see it giving most benefit?
So again, sorry, I was just
where do you see AI technology advancing with regards to your work, and what, I guess, where do you see the most benefit coming in from? What are you most excited about looking a few years down the road?
Yeah, I think so there's actually an issue currently is that we generate a lot of database. Let's see, ChIP-seq database, or, RNA-seq data. So one publication done, you never use that data set, right? So the most exciting things I think coming on the road is how we have a plant science AI knowledge base. We have DNA foundation model, we have RNA foundation. We also can build up a plant, protein foundation. So then all these kind of multi-agent come together, then go back towards, we can actually easily access NCBI, all the RNA-seq data, and come up with some hypothesis. Let's see which gene network, which one is dominant in the gene regulation in response to pathogen. Let's see, right? So we can go back to, transcription factor database. We can go back to all the RNA-seq data set. We can actually starting to faster learning what's really going on. So reuse almost all the heavily data set we already generated. So I think the space on that is very large. So basically, we build up a kind of a multi-agent virtual research lab. So this is my dream. I think it's coming up pretty much like a couple of years later, but we're truly building up. So everything you see here with, DNA foundation, RNA foundation, also the one with the knowledge graph, we're actually building up the loop. So which means that I can, using all these multi-agent to answer a very specific question, I can reason, give actually a hypothesis, then reasoning through the database I already have. So rather than do a lot of experiment towards that, also can come up with a lot of hypotheses which people can actually go explore to design the experiment. So that's something which I can see is actually really can work closely with everyone, rather than, normally people will actually generate a large amount of data, then look into the biology inside. Here is we actually generate a hypothesis. Then we're just using experimental data, to prove whether or not that hypothesis makes sense. So that we're actually changing the direction slightly different.
Great. So I'm being informed that Derek has a question. So I don't know if Derek wants to unmute himself.
Thanks for that. I'll concede some of that was way over my head, but I thoroughly enjoyed it. You hear me okay?
Thank you.
Are you planning this as to remain open source, or can you see routes for commercialization and exploitation?
So currently, plant science AI is not commercially, so it's open source. So everyone, the only thing is you have to registration with your academic email, because we don't want other people to use that if they are not using for research purpose, right? So that is the current base. So plant science AI is open to everyone, and actually you can upload your database. So if you have EndoNote, you can just upload everything. So every weekly base we will upload, we will update the whole knowledge graph. So the node is actually getting bigger. So you have a more understanding of knowledge. So that is continuously building up. And I hope everyone use it, because the more you're using it, the bigger this knowledge graph it is. So far, this is the largest knowledge, plant science knowledge across the world. So we hopefully to keep it running free for everyone. Of course, in long term, I don't know. So currently, is our institute generously funded that. So I don't know how much they continuously fund that, but we want to make it as actually a community resources. For the DNA RNA foundation model, currently we are building something called WUI. So it's a website thing. So you can input your RNA-seq data, the same as you upload your PDF. So we can actually generate that to give you idea your genes, which part of the gene is actually important for expression. So if you upload, for example, 2 columns, one is your promoter region, then your RPKM, we can, based on your upload, give you automatically come up with attention metrics. So we tell you your gene, which part is important to its expression. But however, of course, that pipeline will come, I think, in 1 year a little bit slower than the plant science AI. That is also try to get a whole community effort, is everyone can input your data and you will get an interpretation and everyone can validate afterwards. So we will give you a prediction. We will say, okay, the -20 nucleotide in your translation initiation is actually important for your expression. You do the mutation, then you check whether or not that's the case. We're waiting people to give us feedback as well. because every AI model, you want to reach 100% of prediction, right? You're talking about a maximum like 80, 90%. So that's what we actually want people to use that more frequently. So that's pretty much you can see in the pretty much short term. I think in 1 year time, RNA foundation model, web server, like base things, you can actually get it. I think DNA maybe takes a little bit of time, but I think in 1-2 years time, you definitely will see that coming out from my lab.
It's a great answer. Thank you.
Thanks, Derek. I'm informed now that Raoul has a question. Raoul, if you want.
Yeah. Can you hear me?
Yes.
Okay. Well, fantastic presentation. Thank you.
Thank you.
So I was wondering, you explore the connection between the structure and regulation of the spliceosome with your post-transcriptional regulatory mechanism that is basically focusing the 3D structure of the RNA. Have you explored these connections?
You mean 3D structure?
Yeah, but the structure of the RNA and the connection with the RNA is coming from the spliceosome.
You mean what? Sorry.
Spliceosome.
Spliceosome. Yeah, spliceosome is, so currently we can see, so I didn't explain detail in the spliceosome part. So currently is we measure the pre-mRNA. So the spliceosome whole complex, we didn't measure the substrate inside, but that's a separate thing with cryo-EM people. But the truly is we truly can see the U1 binding site, which actually is important for 5' splice site. So the messenger RNA, so the pre-mRNA, we do see the structure feature, but the whole complex we haven't actually really worked on. So recently, the reason I'm now currently in LMB is actually discuss all these kind of a bigger, cryo-EM structure, whether or not we can capture the bigger structure inside the RNA kind of a structure feature inside.
Okay.
So that's a separate thing.
No,
so
I mentioned that because some time ago we described the post-transcriptional mechanism regulated by the SMA and porcupine, that they have basically the same phenotype that you show us, is core-sensitive mechanisms, and they have early flowering, and they are accumulation of the core layer. And they are core structural element for the spliceosome. So there is a connection there, clearly connection.
Okay. Then we haven't to do that, but that's something. Possibly we will actually do that.
Thank you.
Thank you.
Thanks, Raoul. I think the last person at the moment with their hand up is Run Xuan. So maybe ask Run Xuan if he wants to unmute and ask his question.
Yeah, thank you for this very impressive work, Yiliang. I really enjoyed it. My question is on, you mentioned about for each of the motif you find, there's a 50 million matrix behind it.
52 million matrix depends on your foundation model.
So what is those 52 million?
52 million. So basically is so Reggie, it's actually 52 million matrix is basically just to measure the sequence itself. So 52 million, that means you have pretty much a 52. You think about it more like a token.
All right.
So for example, if I have a CCC here, I have a GGG, they are distanced about 15 nucleotide, I will token it up.
Okay,
if I see a lot of frequency of this sequence crossing the whole, all the training data set. So if there's actually distance of 100, I will build up another token if I see that frequency. So it's basically every single layer is actually a way to token this sequence. So in that way is now I have a CCC, GGG, everything will have a behind itself, will have a lot of dimension to measure this sequence rather than just a CCC. That's what happened. Of course, if you actually pre-training longer time, you will have a more higher matrix behind it. So currently the MOE, we can go for 520 million matrix. It's really towards how many is enough, for learning. I think currently is, it's not required that much. You already can gather some idea.
All right. Okay. So those 52 million is related to the layers and maybe hidden neurons of this network. Okay, I see.
Yeah.
Okay. Thank you. Thank you very much.
Thank you.
Great. Now we've got some questions in the chat here. So I don't know if, so the first one's from, is it Shengji Gao? I don't know if you want to unmute and ask the question, or should we just read it?
I tried to read it. So she had a question is, I have one question regarding to the RNA pre-training expected for the nucleotide sequence? Will the additional information folding structure be reconstructed during pre-training, or they are just being treated as additional input? I needed to say is the folding data structure current version, so the one we published last year, was actually just as additional layer of input. And of course, we currently have a more comprehensive folding. So the previous version is you just have one in silico folding, right? But you think about it, all the structure have a lot of conformation. So that is currently reconstruction for that. So the currently version 1 is only one conformation per one sequence. So as an additional one for the training. So the other question from Elizabeth is, one of the issue talked about regarding AI is the quality of messaging and curation. What is your view on that? I needed to say is a foundation model is relatively tolerant to the data noise. So I give you an idea, right? So if we have two RNA-seq data from two different labs, so when we actually learn the fine-tuning step, we will actually have something called F1 score. So foundation model, you will actually after foundation model, you have these pre-training. Pre-training is typical like an AI step, right? So you have 80% of data to learn, then 20% used to predict whether or not it's learning anything, right? So that will generate something called F1 score. So we will base on F1 score, which one is higher, we know which RNA-seq is likely will generate some learning rule. So this is how we're using the data. But generally, as a foundation model is kind of tolerant to the data set rather than deep learning, which require really good label data. But a foundation, you don't need data sensitive, not like a deep learning. So then there's so Ron had another question is, I was wondering if this research could be applied to the legume focusing on nature genetic variation, especially those crops require tolerance to low temperature early flowering. And I needed to say is, so this is what we currently build. So basically is everyone think about a GWAS, right? GWAS had a lot of limitation is you have so many data sets. And at the very beginning, you have to decide the population, right? So you basically whole genome alignment, you separate different population already. Then you actually calculate something called a selection power, right? So this is actually a little bit biased because at the very beginning, you have to decide the population already separate. So we create a separate thing is for the nature genetic variation library pre-training model, which the first one we're using is Arabidopsis 135. We haven't working on any legume, but we actually working on Wilkinson wheat population as well. So now we independently see all these individual SNP. We don't give any assumption which population it belongs to. So that will actually different than any GWAS currently people do. So we don't know the result yet. It's actually under pre-training now. So we think we likelihood will break the boundary or limitation of GWAS. So that is things hopefully next year you can see some result from our lab. Yeah. So I think I finished everything.
Yeah, that's good. Are there any other questions? If not, can I ask a quick one? So you talked about the plant science AI and the auto SKG. So that's
auto knowledge graph. Yeah.
Yeah. So that's really interesting. We're interested in that kind of stuff too. But what I was wondering about is, so you're updating that with new knowledge as it becomes available. And I just wondered how you deal with a situation where a new bit of literature completely changed the way of thinking. So you might have a group of 12 papers that said something, but there's something new comes along that says something else. And that might be the thing that's correct. How do you stop that from being completely swamped by the older knowledge?
So this part, we don't know how much it is. So I think one of the, paper review, he comes back, he said, you don't have enough literature on the non-seed plant. So when I ask some question, it feels like, seed plant biased, right? Because I don't have enough, literature on the non-seed. So this is, it's hard to say. I think it's really question by question. That's why we provide all the knowledge graph, tell you how we generate this answer. So be able to help you to tracking down how we generated this, answer, rather than just to give you answer. So that's the original idea why we needed to give all these knowledge node together, so you will be able to know whether or not it's good or not. And also second part, very surprising, which maybe can explain a little bit how it is. So you possibly know there's some literature, they spell a word wrongly, right? Error spelling. So actually, when after our learning, we can actually tell you which literature they have a wrong spelling of a certain word. So it actually will tell you, give you idea this is wrong, but the other part seems to all agree with this version of a spelling. It's maybe this is wrong, and tell you which literature may have a problem. So this is give you idea, is it give you option to tell, whether or not I pick up a separate idea in my knowledge. And I don't know whether or not it's right, but I give you idea, I can see the difference. So I think it's truly it depends on how to. I needed to say, when we build up Plant Science AI, we give almost all the feedback link. So if you see anything, you think, can I actually have a more function towards that? We can build up towards that. So we truly find a lot of individual case very diverse. Yeah.
Okay, I just think it's really interesting. It's just an example of why it's nice to see where all the sources come from, and why you shouldn't assume that something is correct just because some AI tool says it's correct.
Yeah. So that's the original purpose why we build up.
Yeah. Great. Well, thank you very much. I don't think there's any other questions. So just I think the last thing to do is just to thank you again, Yiliang, for the talk today. Very, very interesting. If anybody wants to speak to you about stuff, I'm sure they can get in contact with you. We can give out your details and stuff, I'm sure. Yeah. And thanks again.
Thank you.